for that very kind introduction. Thank you to the organizers for inviting me here today to tell you a little bit about my work. Aging. When someone says aging, we ostensibly know what it means, right? It means gray hair. It means wrinkles. It means our grandparents. Aging is so stereotyped that we can now submit a picture to a computer program and estimate how old we are. Or you can even submit a picture of a younger person and it can estimate what they might look like when they're older, like old Justin Timberlake here, for example. And we all age, right? It's inevitable. It's unavoidable. But as my 4-year-old niece likes to say, and as many scientists say, but why? Why do we all age? And as my niece also likes to say, but do I have to? And I think that's a good question. So unfortunately, besides just gray hair and wrinkles, aging comes with a lot of worse things. So for example, cognitive decline starts already in your late 20s, which is quite concerning for people like me in our late 20s. So for all you young people out there, which clearly I am no longer since I say things like all you young people, but for all you young people out there, really enjoy it and soak it up, because it's all downhill from here, fortunately. Now, it gets worse. Aging is actually the highest risk factor for a lot of major diseases, cardiovascular diseases, diabetes, neurodegenerative diseases, cancers. And now, while we usually accept aging as a natural process, we don't accept cancers, we don't accept Alzheimer's and heart attacks. We fight them with everything we can, right? But a lot of these diseases could even be said to be symptoms of aging, in a sense. Again, why? Why is that? And why do different people seemingly age at different rates, right? One of your grandparents may look like they're 50. Another may look like they're 100. Why do different animals age at different rates? If a whale, a shark, if a clam can live to be 200, 300, 500 years, why can't we all? Now, in research, we often use model organisms, like the household mouse. And they're very good models because they're similar to humans in a lot of ways, actually, biologically. But why is it that the mouse's very close cousin, the naked mole rat here, the miraculous naked mole rat, actually lives 10 times as long, or even longer, seemingly without any age-associated diseases? So the goal of aging research is not really to live forever, per se, but actually to live healthier for a longer period of time, right? If we can all grow up to be 122, like the record holder, Jean-Louise Calment here, if we can all be a little bit more like the naked mole rat without these age-associated diseases, hopefully with a little more hair, and if we're really lucky, maybe even make it to 500 and be happy as this clam here. So why can't we all, just with a little bit of help? Now, people have been looking for the fountain of youth for a long time, throughout history, right? Famous explorers, Ponce de Leon looking in Florida, I don't think he found it. Indiana Jones found it in one of his movies, but subsequently lost it very quickly. Even Jay-Z wants to be forever young. He really wants to live forever, forever, and ever. But actually, even though we haven't found this fountain of youth, scientists have made great progress already in slowing down aging, increasing health span in model organisms. So in flies, in worms, in mice, single genetic mutations, interventions like calorie restriction and exercise, and even a few drugs like rapamycin can slow down aging. So there is hope for humans. But back to the question of why. Why do we age? And there have been a lot of theories proposed, reasonable theories and less reasonable theories about why we age. But I think this quote sums it up very nicely. None of them appears to be fully satisfactory, right? We're still looking for answers. However, in contrast, there are a lot of classical hallmarks of aging. Things that are conserved between different animals, conserved between different tissues and cells, that signify aging and maybe contribute to aging. And one in particular that I studied is stem cell exhaustion. So what's a stem cell? In this case, a hematopoietic stem cell, which makes blood. Stem cells have two very defining characteristics. One, they can self-renew, which means make more copies of themselves that normal cells cannot do. And two, they can differentiate into all these other cells in their tissue. So in the case of a blood stem cell, they make all these other mature blood cells. And hematopoietic stem cells in particular are the kind of cells that doctors will transplant into bone marrow transplant patients, right? And we can do very similar experiments in mice. And actually, the experimental definition of an HSC, for short, is that if you take one stem cell, you transplant it into a lethally irradiated recipient, that one stem cell can repopulate the blood system and allow this animal to survive. And so we can use these types of experiments to really test the function and the fitness and the health of these HSCs. So unfortunately, most stem cells lose their regenerative ability with age, like HSCs here. And so when we take stem cells from a young mouse and we transplant them into a recipient mouse, we can take samples of the blood every few months, and we can use special markers to tell what blood cells came from that donor mouse versus the host. And so in a young mouse, you can see on the left in white, that young HSCs really can regenerate the blood very well. However, if you do the same exact experiment and take stem cells from an old mouse, they lose this ability to make blood, significantly worse. And so this loss of regenerative function can directly lead to age-associated blood disorders, diseases. Okay, but why? What causes and regulates stem cell aging? One thing I studied in particular is autophagy. Autophagy is a cellular recycling process in where various contents of a cell are degraded and the building blocks and the nutrients are released back to be reused. Okay, and this is very important for various things in cell health and stress response, but in particular, it's been correlated in a lot of studies with lifespan extension and health span extension. Right, so a lot of these interventions I mentioned actually can induce and or require autophagy. So there's a very strong link there. So for me, one question was, how might autophagy play a role in stem cell aging specifically? Might its beneficial effects on these animals be working through stem cells? So to do this, first thing we did was generate this autophagy deletion model. So we deleted a gene conditionally so that the stem cells could no longer undergo autophagy. We do the same transplantation assay. What we find is that while healthy stem cells can regenerate the blood easily, the cells without autophagy really lose that ability to make new blood. And this is strikingly similar to what we saw in these old stem cells, right? So they have this sort of premature aging phenotype, along with some other things that I'm not showing here. Oops, sorry. So the question is, how then is autophagy affecting this premature stem cell aging? And so we looked with an electron microscope really close at these cells. And what you can see is that in these cells without autophagy, there's this accumulation of these mitochondria. And mitochondria are the powerhouses of cells. They make energy for when a stem cell needs to divide or make new cells, right? And we saw this increased level of mitochondria, increased mitochondrial activity, correlating with increased metabolism and cell division, right? They're proliferating and making more cells. And so this was actually made sense to us because autophagy, as I mentioned, is known to clear organelles like mitochondria, right? So when a stem cell is in its off state, its quiescent state, it has low levels of mitochondria, but we found that when they're activated to make more cells, they'll make more mitochondria, make more cells, and then autophagy will help clear those mitochondria and return the cell back to its off state. However, when we deleted autophagy, there was a loss of this ability to return to the off state. They're stuck in an on state with too many mitochondria, too much cell division, and then eventual stem cell exhaustion like we saw. Okay, what about aging? How does that affect aging stem cells? So next we looked at old mice, and again with this electron microscope, and we looked very close, and we saw that while most old stem cells didn't have any signs of autophagy, there was a small subset of about a third of the cells that had this high level of active autophagy recycling. This new subpopulation we didn't know existed until now. And so we used a special mouse, a GFP-LC3 reporter mouse, which specifically can show us how much autophagy each stem cell is undergoing. And so we use a machine where we can separate these two different populations so we can study them separately. And we go back to this transplantation experiment, right? And if we put the cells with low autophagy, old cells with low autophagy, transplant them into a recipient mouse, they initially can make a little bit of blood, but over the course of 4 months, they're really exhausted and tired. And if we look at the stem cell compartment specifically, after 4 months, they're almost completely gone, depleted, exhausted. However, when we took stem cells from the same mice, the same old mice, but that had high levels of autophagy, suddenly they were able to regenerate the blood long-term for 4+ months, and the stem cells at the end were all totally intact and healthy, still functional, still there. And so back to this model, we found that the majority of old stem cells do indeed become dysfunctional as we knew, and they resemble these autophagy-deficient stem cells. However, we discovered this population, this rare population of old stem cells that use high autophagy, high recycling to stay healthier and younger. And when we looked at various other characteristics of stem cells, they looked identical to a young stem cell in everything that we looked at. So I was interested, of course, in if this might be conserved in other systems and other tissues. And so we looked at mesenchymal stem cells, neural stem cells. We found very similar results. Now, what's the impact potentially for humans and aging and disease? We also found that the stem cells with low autophagy had these signs of gamma H2AX replication stress, DNA replication stress, potential DNA damage. And the cells with high autophagy had almost none. And so this could actually lead potentially to something called clonal hematopoiesis, which was recently described a few years ago, that occurs in elderly people. Now, clonal hematopoiesis is when instead of each of your stem cells making a little bit of blood evenly in a healthy manner, a few of your stem cells are making a whole lot of blood and the rest are not doing much at all. So there's this even production of blood. And so you can imagine these stem cells with low autophagy that have higher and lower regenerative potentials. This heterogeneity could lead to this clonal hematopoiesis. Why is that important? Because actually the clonal hematopoiesis was also found to be very highly associated with increases in blood cancers and general overall mortality, right? So there's this very strong link to aging and mortality and death. Okay, so to summarize, we found that while most old stem cells, hematopoietic stem cells and others, have these low levels of autophagy and are much less healthy, lose their regenerative function, there's this subset that we discovered that have high levels of autophagy that resemble a young stem cell in every way we looked. And so the next question really is, can we make more of those, right? Can we convert the unhealthy cells into the more healthy functional cells? So this brings me to a second project that we're working on now with the idea of rejuvenating stem cells, right? With these various interventions that I mentioned, calorie restriction, exercise, and some others, to see if we can convert more of these unhealthy cells to healthy cells. In one example in particular, something we've working on, is something called parabiosis, which is very popular these days and a lot of really exciting research you might have heard about. But in this process, you take two animals and you stitch them together surgically, and their blood systems are joined. So what happens, I know it's a Frankenstein-like experiment, but what happens is these aging factors and these youthful factors will mix between these mice, and what a lot of labs have now found is that these youthful factors actually will make the old mouse a lot healthier in a lot of ways. They'll be smarter, they'll have stronger muscles. And so what we're studying is how specifically these youthful factors might help an old hematopoietic stem cell that actually makes blood. Now, after grad school, I actually wanted to switch and try something very different, neuroscience. I think it's important to challenge yourself, to learn new things, to succeed in different environments, right? Get out of your comfort zone. And the brain, I think, is just a miraculous organ. It makes you who you are, holds all your memories, and it's so complex in everything that we do every day, but there's a lot we still don't know. So I've switched over now into neuroscience, and I'll give you a few examples of the kind of things that we're doing in the lab and the technologies that are really amazing these days, that really enable great science. So in this example here, called tissue clearing, what we can do is, through a chemical process, make a normally opaque brain, a mouse brain, entirely transparent, right, as you can see in the top left. And so now what this enables is imaging the whole brain intact throughout the entire thing. So we can see neurons in the deepest part and the bottom of the brain. This is really useful for mapping brain-wide neural connections for various diseases and behaviors. Another really amazing thing that we do a lot is something called multifiber photometry. So in this case, we implant fiber optic cables into a mouse brain. They're very fine, they're healthy. And you can track that neural activity live as these mice will move around and go through a maze or do some kind of task through all these different regions simultaneously. Then what we can do at the same time is something called optogenetics, where we use light to turn these specific regions on or off as we need. So you can imagine a mouse is going through a maze, you're seeing what it's doing neurally, and then you can just turn certain regions on and off and see if it has better or worse memory, right? So this is basically the closest thing we have, as far as I'm aware, to mind control. It's pretty amazing. Finally, we can now have single-cell resolution for live imaging of animals, of mice, while they do behavior. So we can use a microscope and look really, really close. We can see individual neurons firing and activating as a mouse does some behavior, like licking for food. And then, again, we can use this optogenetics to specifically turn on one cell, turn off another cell, very quickly and very precisely. So this allows us to really control behavior, again, with a single-cell resolution and see how it affects this mouse as it does behavior. So with that, I'll leave you with a few final thoughts. Aging is the greatest risk factor for almost all major disease groups. And it's something we can study and something we can understand and hopefully can leverage to help advance human health, right? We found that there's a novel subset of these stem cells that maintain function as we get older by activating and using this autophagy recycling process. So there's actually good hope for stem cell rejuvenation, for slowing down aging eventually in humans even. Generally speaking, I think science is amazing, right? There's nothing I'd rather be doing. The idea to be able to go to work every day, to try to answer a question that no one has figured out yet, it's really just a privilege. And so for young people out there who haven't started your cognitive decline yet, go find an internship, go volunteer in a lab. I don't think you'll regret it. And finally, a lot of my friends will often ask me, I'm already starting to forget things. What can I do to slow down aging? How can I avoid Alzheimer's? And we don't have these answers yet, right? But there actually are a few things I think that we all know are good for us and actually are really good for anti-aging, for diseases, general health. So exercise, aerobic exercise is just amazing. Amazing for your brain, amazing for your whole body. Eating healthy as much as you can, it really is important. And finally, because we don't have this magic pill to cure aging yet, life is really short, right? We could still be hit by a bus any day. So you really gotta enjoy life to the fullest. Enjoy your youth, and buckle your seatbelt, go running every now and then, eat healthy when you can. And as Adam Sandler said in Billy Madison, cherish it, right? Cherish it, cherish your youth. And finally, the most important slide, the acknowledgement slide. I have to thank, of course, my great mentors, advisors, my former advisor, Emmanuel Patzigay, my current advisor, Dr. Carl Diceroth, all the fantastic colleagues and friends and advisors that I've had, my funding sources that allowed me to do my research. Thank you for your time and attention.